Peter's the section chief on functional imaging methods in the Laboratory of Brain Cognition at the NIMH. But of course, he's better known as a walking Wikipedia of fMRI. And of course, as one of the first three fMRIers for his thesis work in demonstrating bold fMRI. He was at the MGH NMR Center from 1994 to 96. So I just missed, I think, overlapping with him here. My loss, because of course he's gone on to have a stellar career at the NIH, and more than anyone else in the field, I think he has his finger on the pulse of what's happening in the functional imaging world. So I'm always looking forward to hearing what he has to say. Thanks, Peter.
Thanks, Larry, and thanks, John, as well, for inviting me here. Okay, good. All right. Yeah, it's quite an honor to be here to be giving a talk in the context of 7 years of fMRI, or 7. God, that was such a mixture of all kinds of things. 20 years of 7 Tesla fMRI. Wow. And yeah, it's really great to come back. Whenever I come back, it always feels like, in some ways I haven't left. In some ways I haven't really left. I'm still friends with so many people here, and it seems that so many things have changed. I left before the onset of the 7 Tesla, but at the same time, things have sort of stayed the same in a good way. So, all right. So what I'm going to talk about today is my work at the NIH and how our scanners have sort of a little bit of the history, a few pictures here and there, but mostly about my perspective of what the 7 Tesla can do, as far as that's concerned. So this actually goes back to 1987, way before I was at the NIH, and just to show how far we've gone. This was an article from the NIH in which they talked about their 7 Tesla, their 10 centimeter 7 Tesla spectrometer. So that was state of the art at the time. You might recognize a few faces there, and that's pretty much when the NMR Center started in 1987. So since then, it's obviously been growing ever since MRI has been so successful. This is just a quick history of not all the scanners at the NIH, simply the scanners in the fMRI Center. And without going into too much detail, just wanted to emphasize that we had both GE and Siemens 3 Teslas. And then we finally switched out our 3 Tesla for Siemens 7T. Well, this was our second 7T. The first 7T came earlier than this. That wasn't part of the NMR, it wasn't part of the Functional MRI Center that Jeff Dunn will be talking about a little bit more. But then also recently, we just got a new 7T. The NMR Center got a 7T, the Siemens Terra, in 2019. So now we have 2 7Ts. And this one actually is unique in the sense that there's a picture of its delivery. And it was the biggest crane I ever saw that actually brought this in. One thing unique about this is that it was the first actively shielded 7 Tesla scanner, which was really nice in the sense that we didn't really have to apply all that iron all over the place. And we just had a little bit of mu metal along the walls just to contain the field in just enough so that the hallways were safe for people with pacemakers. So that was unique. There's the picture of it. And this is sort of where it's placed in the NMR Center right now. This is where our 7T is right now. And the old 7T was replaced with the 11.7 scanner right there. And since then, we've sort of added a lot to it. We have up to 8-channel coils and not 32-channel or 32-channel coil right here. We have many different subject interface devices and so on, the standard stuff. So 8 months after that delivery, we finally got our first images, December 13th, 2010. Sean Merritt was our first subject, and it's interesting. He also reported, as you're going into the scanner, he reported, sort of sensing this sort of vertigo, sort of twirling around the scanner. And I think that since then, not many people have reported that. We probably lowered the speed of the scanner going in. So we finally got our first anatomic and functional images, December 13th, 2010. And it's interesting, sort of coincided about with Sean Merritt's birthday, his 50th birthday. So my wife decided to make a cake, a Siemens 7 Tesla cake. So that's pretty much Sean blowing out his Siemens 7 Tesla cake on his birthday. So one thing I have to emphasize about this 7 Tesla is that it really does take a team to keep this going. It's not, it became more turnkey, but it was required a lot of input, a lot of debugging, a lot of debugging of programs, a lot of working with hardware, and all kinds of, at every level of, not quite the level at what was done at MGH, but Martinos Center, but still required a team. And so this pretty much so between Sean, Lin-Ching Li, and Rup Chen Singe, and Andy Derbyshire, they sort of had been handling the 7T, along with the other scanners as well. But and they really have been busy, sort of trying to keep it state of the art. And what's interesting is that it really has caught on. There's a lot of researchers at the NIH that care both about high resolution and either high speed or a little bit more sensitivity, and also structural imaging. And what's interesting is that if you look at, this is different times of the week. So this is sort of a busy bar chart comparing across 2016 to 19. The 7T is used just as much almost on average than all of our 3Ts. And in fact, it's even used a little bit more on weekends than all of our 3Ts. So it compares well. There's a lot of users who are finding it useful enough and important enough to actually address their questions. All right, and this is, so Renzo Huber was a postdoc in my group. I'll be showing some of his work in a few minutes, but he actually keeps a chart online of all the 7 Tesla scanners in the world. Right now I counted 82. And they're pretty much distributing themselves around Europe. There's many in Japan, Korea, South Korea, and China. And they're starting to populate, I was actually kind of surprised to look at this chart, how many are all over the place. And it's interesting too, is that there's real clinical applications. It's FDA approved. So the very simple diagram of what I see as the advantages, it's the higher signal to noise and higher susceptibility contrast, which includes functional contrast in that regard. Challenges, of course, you have RF heating, RF inhomogeneity, B0 inhomogeneities, shorter T2 star and T2, longer T1. But you also have, not only with longer T1, you also have, this is some just work reported that not only does T1 get longer here, but it also gets a little bit more broadly distributed, which can potentially be looked at as both an advantage or disadvantage. I mean, it's interesting why you might get this distribution of T1 or T2 as well. T2 also becomes more broadly distributed and homogeneous as well. But in spite of those disadvantages, we're still addressing those. There are many users who use the scanners, who use the 7 Tesla. Not all of these people use our 7 Tesla, but there is a large fraction that do. And adding up all the papers, there's about 60 papers published using our 7 Tesla over there since the second 7 Tesla, since it came in. And I would have thought that fMRI sort of would have been the dominant thing initially, but really structural applications, mostly MS. With Danny Reich, MS Research, has been the dominant use of 7 Tesla. A lot of method development still, and a little bit of spectroscopy and some fMRI. And just to quickly go through some of the structural imaging, first of all, this is some work by Danny Reich's group showing that you can easily see, much more so than at 3 Tesla, you can see cortical lesions in MS. And this doesn't show up that well there, but there's several different types of lesions with MS that you can actually characterize all individually at 7T where you couldn't before. And this is actually in collaboration with Jeff Dunn's group as well. What's interesting is that there's a specific type of diagnosis now that they're coming up with MS, and that's a central vein diagnosis, where they actually see, based on its susceptibility, the central vein, and also an iron rim around these sort of hyperintensities. And you could also see cortical lesions here. What's interesting too is that it's a differential diagnosis. So this is a study where these are the indicators of MS. This is sort of a general brain inflammation with a pituitary dysfunction. And Danny Reich actually showed that almost actually with only a small subject pool, 17 subjects, 100% differentiation between non-MS and MS patients. So it's a really powerful and precise differentiation. And you can even do it, this is comparing with gadolinium, without gadolinium. You can even see pretty easily the lesions without gadolinium. Okay, so moving on, basic functional imaging. So this chart is really one of my favorite diagrams. Christina helped pioneer this, Gunnar Kruger before that. And what's really nice about this is that it really shows our challenge. I mean, there's physiologic noise really starts to kill us at higher signal to noises. However, just to talk about basic fMRI, even at these low resolutions where there's a high signal to noise, there's still higher contrast to noise, and there's still room where it actually is still even better at these lower resolutions at 7 Tesla. As demonstrated by this paper from the NIH, actually by Sam Teresi, just showing a simple cognitive comparison, cognitive study with 2.5 millimeter voxels with a go, no-go task, essentially. And you can see pretty clearly that the percent signal change and the t-statistics are pretty strongly superior with 7T. And this is with the no-go. You can see all kinds of areas that show clear, much higher percent signal changes, much stronger t-statistics. And that's just the difference between the two maps, all kinds of areas that you wouldn't see otherwise show up. And you can also look at, you can also make very small seeds for regions of interest that actually, you can do basic resting state connectivity studies that you couldn't do at 3 Tesla. So this is basically looking at the connectivity of the bed nucleus of the stria terminalis. And that's right here, just a tiny region of interest. You actually can see this subcortical connectivity much more robustly. And other additions onto this are the use of trying to gate, multi-echo, things like that to even get it a little bit more stable. However, 7 Tesla really does differentiate this nicely where 3 Tesla wouldn't. Okay, now to get to the main advantage, I think, of 7T for functional MRI at least. And that is going to extremely high resolution. So what's nice about high resolution is that it sort of bridges this gap between genetics, single neurons, spatially limited calcium imaging and so on, all the way to larger fMRI cortical regions. So you can actually start to get at the functional units of activation at the layer level, at the column level. And so why is this important? Well, from understanding the literature, we can actually try to make assessments as to what the cortical input is, what the outputs are of these columns. And it's certainly complicated. This is just one example from Feldman and Van Essen showing a layer-specific diagram. But if you can actually start to isolate what areas, what layers are activated, you can start to draw connections, not just draw arrowless lines, but you actually can start to draw connections as to what feeds into what areas to get at causality more directly. And the person who's been pioneering this has been Renzo Huber in my group, who's been really a force of nature in some sense, doing blood volume imaging to look at layer-specific fMRI. So what do you need for layer-specific fMRI? You need small voxels below 1 millimeter, preferably about 0.75 millimeter. You need high sensitivity. You need at least a signal to noise of 20 to 1, which would average up to about a signal to noise of 40 to 1. You need high specificity. You need a pulse sequence that doesn't suffer from large vessel effects. Vaso, which is sensitive to blood volume, seems to be the best, but it's not clearly the best. It's in the lead. And you need the right paradigms to obviously tease apart layer activation. So once again, looking at this diagram here, looking more down here, this is really a sharp cutoff. And this happens to be 1 millimeter, but if you go a little bit further down, you start to get into this area where it makes a difference between averaging, for a half hour versus averaging for 3 hours, which is undoable, right around here, especially when you're looking for a 1% signal change. And 7 Tesla is really critical along this steep part of this curve for those resolutions at just below 1 millimeter. And this is just taking that a little bit further. If you're starting with a signal noise of 20 and looking at a effect size of 1%, you're just about able to see it after averaging for about a half hour. So you're really at a very, very sharp cutoff at that steep part of that curve. And where 7 Tesla is critical for seeing this layer-specific activity within a reasonable amount of time. So as far as specificity is concerned, I won't go into detail with this chart, but essentially most of our pulse sequences, most of the contrasts sort of fall along this sensitivity specificity curve, but vaso, which is sensitive to blood volume, seems to pop out a little bit in terms of sensitivity for the specificity that you get. There's other sequences that are in a running, but vaso seems to be pretty good as far as that's concerned, which I was surprised with. I actually initially thought vaso was a little bit more weighted towards larger vessels, but it clearly seems like it's more sensitive to small veins or capillaries and not the ascending vessels. So the concept behind vaso is very simply, you give an inversion pulse, you wait until the blood signal reaches a null point, and then you can collect here, or invert again and collect another bold signal for the same, for essentially for free, and collect them both in parallel. Where you have the blood signal is nulled, and therefore an increase in blood volume would cause a decrease in the signal. And it seems that the changes that occur in specific layers, your smaller vessels, are more sensitive or more apparent with vaso than with bold. Okay, so very quickly I'm going to go through, so essentially he's demonstrated this many different contexts. One is using different types of finger tapping paradigms where you tap with touch, tap without touch, touch only, and ipsilateral tapping. And the idea is that with tapping with touch, you have cortical input and spinal output. But if you have tapping without touch, you have less cortical input from the sensory cortex than the motor cortex. And that's actually what we see right here. Tapping with touch are these two humps with the upper layers and the lower layers becoming active. Tapping without touch, it's damped. Touching only, you only get the sensory input right here and nothing in the output. And then ipsilateral tapping, it seems like you even get an inhibition coming into the upper layers. If you look at bold, you kind of see hints of this, but it's highly weighted by the Peel vessels. We also could do resting state with this. So if we take regions of interest in the upper and lower layers of the motor cortex, we can find that it turns out if you take the superficial layers, which receives input, you have input from the sensory, mostly the sensory cortex. If you have the lower layers, which give output or receive input, we have premotor cortex as well. So it's very different cortical areas depending on which layer you take. And if you flip it around and take the regions of interest in the premotor seed versus the S1 seed, you can get a very different layer profile in M1 based on what seed you choose. So it kind of shows it both ways. And we commonly see this. We see either a double peak showing areas that are lower in the hierarchy and single peaks that are areas that are higher in the cortical hierarchy, it seems. We're still trying to make a little bit more sense of this, but it's kind of the early stages of just looking at the data. Not only layers can we do, we actually have kind of made in my sense, it's sort of a new finding, and that's with looking at digits. With finger tapping, everyone thought that the standard homunculus is, with motor cortex, as with sensory, it's sort of, thumb to pinky finger, which it is in the sensory cortex. But it turns out, looking at motor cortex, there's a mirror image organization, just like between V1 and V2, that it goes index to pinky, or pinky to index, index to pinky, and it even flips again a little bit later along M1. And what it turns out that this flipping is exactly corresponding to the macroscopic difference in activation with either squeezing or expanding. So a squeezing has a different digit map than expanding your fingers right here. So that seems like a new insight into cortical organization. All right, we've expanded this to cognitive tasks. Looking at dorsolateral prefrontal cortex. Emily Finn collaborated with Renzo Huber to do this task, which we're pretty excited about as well. So basically, it's known that dorsolateral prefrontal cortex is known to be active during working memory. We just wanted to find out specifically how it can be modulated. And these are the areas that we found. It's extremely intensive trying to look at the surface and then average along the surface to get the activation. And these are our localizers. These are our typical scans right here. And in a summary, this is what we did. The task was, yeah, a couple more slides, but that's about it. The task essentially was either a working memory task where they looked at digits and just held it in their mind, or manipulated the digits, and then either responded or didn't respond. So it's not a go, no-go task, but it's like they were told to respond or not respond. Looking at the upper layers, you can actually see that the maintenance was higher than manipulation, or the maintenance was lower than manipulation. So manipulating the task caused more activation in the upper layers, whereas in the lower layers, this wasn't modulated by this task, but it was much more modulated by the response or no response. So you can actually even map this out here. The upper layers are in green, lower layers in red, pretty easily seeing the difference in either memory maintenance versus response. Okay, and lastly, very quickly, we're sort of exploring visual cortex hierarchy, and we've looked at 3 different regions of interest, LGN, V1, and MT. And what we've done is, and this is actually localizing, and this is just purely our echoplanar vaso-based images, where we can actually even see the stratigenary in these images themselves. And what we found is this, is that if we look at resting state, and we look at, we take a seed in MT, and then look at the profile, the resting state connectivity profile in V1, we can see that with MT, you have this double hump sort of profile. Whereas if you take LGN, which is lower in the hierarchy, and you take a seed there, you see this single hump profile. So very different profiles depending on where in the hierarchy you're looking. So areas that are higher in the hierarchy than the area that you're looking, you see this double humps. Areas that are lower typically show this single hump. I'm sure it's not as simple as that, but this is typically what we see. And so basically, the idea is that this receives feed forward input from LGN, this receives feedback from MT. And if we take it a little bit further, this is basically looking at LGN. You see, if we color code the seed profile, so this is the seed, we color code the layer profile such that we either get this single hump or this double hump. Depending on where you are, the profile look very different. So this is a seed in LGN. Everything looks like this. Everything is essentially above LGN. But if you move to V1, you see that only most areas are above, but some areas are below. And as you slide that seed along, you see that the profile changes completely, but MT never changes. So you get a sense of, you can potentially get a sense of cortical hierarchy based on what the profile in the layer is to the seed chosen in resting state. Okay, lastly, not going to go into this too deep because I don't have time, but essentially we're doing, looking at the primary energy across each layer, across each layer in columns across the brain. And we see a very clear differentiation between anterior and posterior parts of the brain in terms of what the layer profile is of the energy contained in resting state, which we don't completely understand. It could be due to cortical thickness or myelination. And lastly, we're trying to expand this now. So Renzo is coming up with we're having a big limit in vaso is you're limited to a slab. And there are strategies that one could use to actually do whole brain vaso. And the goal then is to, rather than just make connectivity maps, you can make layer-specific connectivity maps across all different regions. So we're just beginning to try to figure out the connectivity profiles across these areas. But essentially, that's the goal, to actually make, rather than do just region of interest space layer connectivity, have that extra dimension of looking at across layers across the whole brain connectivity. And then lastly, I just want to end with some sort of, a little bit of an intriguing sort of thing, is that we've worked with another vision scientist at the NIH who claims that he was comparing vaso directly with BOLD. He claims that BOLD is actually much better than vaso for detailed general activation discrimination. So it's a higher contrast to noise. You don't exactly know what the weighting is across layers, but it doesn't really matter in this context, whereas vaso is much better for layer-specific discrimination. So it's something to think about, at least if you're doing detailed retinotopy or other sort of detailed mapping as opposed to specifically layer mapping. All right, with that, just like to end, we have lots of collaborators, lots of people that we're working with. Renzo also keeps a list of all the layer papers online. If you want to check that out and keep on top of it. Most papers have been published since 2016. And also just want to end with one last slide, a challenge. What I see as the main challenges for the future of 7 Tesla is we really want to get capillary sensitive whole brain fMRI, which vaso seems like it's moving in that direction. We still have a long way to go for full removal of physiologic noise. We also we're moving towards increased RF homogeneity and efficiency, and then simultaneous EEG and fMRI at 7 Tesla is not trivial. There's a lot of issues that need to be overcome that have been already at 3 Tesla. So with that, thank you.